Let's get started. So this is week 13 and the last week for the lecture. So I'm going to go through networks or graph. So the most important concept in systems biology. When you study a system as a whole, you need to understand individuals. And most importantly, the individuals are linked. They influence each other. So anything related to the system, and you need to understand it from a network level, okay? So that's all the systems biology is doing, and all the systems biologists consider not only the single identity, not only one gene, one cell, one organ, they have to consider the interaction, the relationship among multiple items at the same time. So that's why network is very, very important if you want to study a system, and we put in the very end of the lecture theory. If you remember, we do mention this PageRanker algorithm. So in the first lecture about machine learning introduction. So PageRanker is a network theory measurements or index to measure the importance of individual nodes in the network. So this is like a full loop back, full circle. We end with the network introduction. So I will mention this definition of graph network. You will realize again everything is a matrix. So throughout the whole lecture theory, matrix, I discuss all the time. And every algorithm, no matter how simple, how complicated, even for this language model now, it's all about matrix, multiplications. There are a lot of matrix again. So in the network theory, a network itself is a matrix, for example. Then we will discuss if you have a matrix as a representative of the network, the relationship between nodes, what you can do with this. So first thing, we will have centrality measurements that tell you how can you compute the importance of individual nodes to use a number to characterize the nodes features of importance in the network. Then we will discuss a method called WGCNA. The reason I put in here is it's a network method, and to help you to understand the importance of each node. For example, a gene-gene interaction or co-expression network. This method has been developed and become very popular in the gene expression analysis field. In terms of gene expression analysis, the first thing you will think about will be differential expression, so high, low in different conditions. If you move forward one step away from the simple DE analysis in the bulk RNA, before single cell become available, gene expression analysis will mostly end with WGCNA. It's so popular, maybe cited for thousands of times. So I have to mention this method so you know it's there and the principle behind this. People still use this for single cell data analysis now, but it's not mainstream now. So you understand how it goes and how the algorithm is designed. So next time when you decide to use it, WGCNA, follow the popularity because of the momentum. The reviewer may ask you to do the WGCNA co-expression analysis. You know what you are doing. not recommend. It's not the best algorithm anyway. So we will introduce very quickly the module detection. So from the communities in the network, then I will move on into the single cell data analysis for gene regulatory network analysis. So this is to use single cell data to construct gene regulatory network. And I will mention the Gene3 as one of the important developed tool that can derive single cell gene regulatory network very accurately and according to the golden standard. So we have some of this small scale gene regulatory network. We know this method worked. Then this is the algorithm behind Gene3. Okay, this is a classification or regression tree based. Then we will mention the multi-layer. So if you have a time series networks, what you can do, and we don't have a good solutions yet, but conceptually, we should be able to think or consider which direction we should put forward effort on. Then at the end, I will introduce the single cell 24-net XCT, the tools we developed in our lab. Okay. So I don't know you have done this before, but in general, you want to create a correlation matrix. So this is an example I get from a brain study. So the neuroscience people, and they measure different location on the brain. Okay, you can have. So this is MRI. You can have EEG signals. So you collect samples and across time for each location. So here you have orange, and then you have this blue. Signals can be recorded at the same time, right? So if you have this signal and that signal, you can compute a correlation between them. And if you have more than two, and you have, see, okay, 50 or 100 sample points, and you can create this 50 by 50 or 100 by 100 correlation matrix between pairwise correlation between each sample points. So that's an input for our network analysis. So this can be considered as a network. You can convert this using some filtering and to create this adjacency matrix. Adjacency matrix is network. So once you have this network, you can start the function link. Say, okay, so this region always fired up at the same time, and another region, although maybe very close to each other, never co-expressed some pattern from there. So from this step to functional analysis for the network that rely on the algorithm. So this is one of the example in the neuroscience. You can generate network using this method. I want to quickly mention the relationship between correlations and regression. So the example I show here is correlation matrix. Basically, if you have signal A, so in this case will be the gene A expression across cells, gene B expression across cells, you can compute a correlation, right? You can also build a regression between these two and use, for example, X, use the gene expression X to explain the gene expression in gene B, Y. So this is a linear regression relationship. And then for this, you will get beta, which is a slope of this. So you can use either R, this correlation coefficient, to build the network, okay, to construct the link between two signals, two genes, expression profiles. You can also use beta. They are related. You can convert R coefficient with the standard deviation, this ratio, and you compute the beta. So that's just let you know. Sometimes people use co-expression, they use correlation. Sometimes use slope, use beta, this regression coefficient to get this matrix. Okay. So that's the definition of matrix, of adjacency matrix, to describe a network or graph. So in graph, we talk about edges and the vertices. Okay. In the network, we call the link or nodes. They're all the same. For this network, for example, you can there's no direction. So 1, 2 are linked, 2, 1 are linked. So that's one. The index 1, 2, 2, 1 are all ones. And if you 1 and 3 not linked, so the position in this matrix 1 and 3 is 0, 3 and 1 is 0. Okay, so this adjacency matrix. I hope nobody have any problem for this, right? Nobody, for sure. So if I have a network, of course I will give you a matrix, and if you want to visualize it, you can draw a network like this. So what's the first thing you want to do, or you can do, is to look at those nodes and which one is important. The simplest and straightforward the measurement is to call degree. Okay, you look at the nodes in your network, how many direct link you have. Okay. For instance, in this example network, the purple one has 4 direct links. So it has a degree 4. Okay. 1, 2, 3, 4. And if you look at this, it's like empty. There's only one link. So degree for this node is 1. Well, this node is 1, 2, 3, 4, 5, 6. Okay. By simply just measure the level of degree, I would see in 80% of times you are fine. The highest degree indicates the important node, the importance level of the nodes gave you. But there are hundreds of other measurements has been defined. Okay, I see 100, maybe over 100. Okay, if you look at the literatures. So those measurements are called centrality. So centrality means how essential or how central, how importance of each node in the network. Oh, here is your question.
I have a question regarding that figure over there.
Which one? This one?
Yes, that one.
Blue one.
Since written degree equals to 4, and from what I understood is degree is the number of connections a node has.
Yeah.
Then how is it 4? Okay.
So 1, 2, 3, 4. Yeah, purple one has 4. So this blue one has 2. Yeah. Yeah. So the network theory people realize only rely on degree sometimes not sufficient enough. For instance, if you consider the blue ones, seems not very important, but if you actually remove this, it will break the communication between these two subnetworks. So like this is in the kind of a bridging point. So in some sense, this blue point is more important than others with a higher degree. For example, this has a 3 degree, but maybe not as important as the blue one, only have 2 degree. So that's why a lot of this centrality index has been developed, proposed in the, definitely not only in the biology field, in the social network, in the all the network theory field, people develop different kinds of centrality measurements. Give you an example is to it's called closeness centrality. So if you look at this this network, okay. So look at that. If you look at the B, have a degree 2, C degree 2, D degree 2. They are all the same. These 3, right? Of course, for the degree level, so the A and the E is less important. But for closeness centrality, consider the it's 1 over the total distance from the node to other node. For instance, if you compute the closeness centrality for A, and then first you will look at A to other nodes, the distance. So now the distance is by steps. To B is 1 step away, so distance to this one is 1. So you put 1 here. And from A to C, you need 2 steps, 1, 2. So put a 2 here. Okay. For A to D, you need 1, 2, 3. So that's 3. From A to E, you need 4 steps. So you put all the distance together, sum them, then take the inverse. Okay. If you have to move multiple steps to your neighbor, and your total will be a large distance. So your centrality should be small because you are away from the majority. But if you look at the C, it's somehow in the center. It moved to all the neighbor nodes or other nodes in the network in a relatively short distance overall. So your total of this distance summation will be smaller, so 1 over it will be larger. So that's why if you compute the closeness centrality for C, this is a 0.16. It's higher than D and the B. Okay, so this is a very simple example to give you a counter example, which although B, C, D have the same degree, and if you measure the importance, use closeness centrality, and the C seems more important because it's located in the majority center of the network. So people also use not a distance, but it is called a harmonic mean of all the distance, harmonic mean. So that's 1 over the distance. So for this, for example, the distance from this to others will be 1, 2, 2, 3, 4. But instead of sum them together, and you get this. Convert the distance into the rate. Okay, so sum this to use this as a centrality measure. So before move to another centrality introduction, I want to show you this. It's a very useful concept, and I hope you are still focused enough to get the idea of this. Okay, so now we are talking about directed matrix, directed network. Directed means you have the distance, you have steps, and also the direction. So, for example, 1 to 3, this is the arrow, so you can move from 1 to 3, but not 3 back to 1. So if you look at this adjacent matrix, this location, which index 1, 3 is 1. You can move from 1 to 3. You cannot move from 3 to 1 because the 3 to 1 location is 0. Okay? So for directed graph or directed network, this adjacency matrix is not symmetric. If it's symmetric, it means you can go either way in the same direction this, okay? But this is not. So we have the translation of this directed network into a matrix, adjacency matrix M, okay? So if you don't understand the check, so it's 1, 4 is linked, 1, 4 is linked, 4, 1 is not. And here is 3, let's see, 3, 3, 1, yeah, 1, 3. Okay. Also, this is a squared matrix because you consider O to O link, right? So now we are going to do matrix multiplication. So this is M. M is n by n. Now we are going to do is m by m. M times itself. Or you can write so m power 2. So this is a matrix. Matrix multiplied with matrix. First thing we need to check, can you do it? You can, because this inner dimension are the same, so m here. Right? So that's the way I show you. You need to do this. So it's every row, column, and multiplicate. And here the result is do the multiplication squared. It's n by n, n by n. The result is also n by n, right? My question is, can you do one more? So this dimension is n by n, so you can keep doing this. You can multiplicate this again with another. So if you have m squared, and you can multiply it by another m here, you can m. They all n by n, so we can keep doing this. So our question is, so you know how to compute this. You extract one column here and one row here and compute the column here, so to fill up those positions. That's the way you compute. Basically, the dot product between column and rows, okay? So for our example, if you do m times m, you get this. Okay, you can check, use MATLAB or use manual check. Now, so what does that mean here? So this place, for example, so this place becomes 2. So this index of this is 2, 3. Okay, I'm talking about this particular location as an example. Okay, I did adjacency matrix M multiplied by itself and get a adjacency matrix squared. And get one of the elements in location 2, 3, and it become a number 2. So what that means is this number indicates from node number 2 to 3. So that 2 to 3. You have two different ways to travel from a 2 to 3. You have two paths to go from 2 to 3 by step 2, using 2 steps. So I show you the steps. So from 2 to 3, you can go, there's no direct link. If you look at M here, 2 to 3, it was unlinked. There's no directly parse. You can go one step directly. But if you do this power 2, this tells you if you allow me to do 2 steps travel, I have 2 different ways I can travel from 2 to 3. So the first one will be to go to this and that. Okay, this is my first. My second will be going from 2 to 5 to 3. Okay, so this number tells you how many 2-step paths you can go travel from one node to another. So now interpret this. So the 3 means from which node? It's number 4 to 1. So from 4 to 1, so from 4 to 1, 4 to 1, 4 to 1. So from 4 to 1, there's no direct link. But if you consider 2 steps, the power 2, you have 3 different ways to move. So I'm not going to draw it because it's too complicated. So just remember, we will try to move from 4 to 1. So the first part will be to go 4, 5, 1. You can go 4, 6, 1. You can also go 4, 2, 1. Okay, so that's why you count 3. There's no more. So now you understand the powers of adjacent matrix, what that means. So same thing. So once you see this, I can give you to the power of 3. Okay? So the result you will get will be how many links between 2 nodes can go 3 steps, how many different ways you can travel.
Also one node is also linked.
Uh-huh.
Also one node is also linked. Like for example here 3 to 2, I have one node jump. I mean one node walk. That is also there in this M square.
3 to 2, so which one? This 3 to 2 not linked.
No, 3 to 1. That will be 3 to 1. The other one, 1 1 in the second column. Second column, third row.
Which one? Second column. You come to here point.
Sorry.
Yeah.
So will be this, right?
Yeah, 3 to 2.
And the same will be there. So it's like it will show 2 jumps and also 1 jump.
I don't think so. If it 2 jumps, that means you only have. So this only shows 2-step path,
Not 1.
not adding this. So if you want to include both 1 step and 2 step, you have to add this to that. This plus that. So that allow you to do 1 step or 2 steps. Yeah. That's a good point. So this 3, 2, so it's 1 step. You can also go 3 to 2 by through 4. So you can expand this idea. If you have a 3, that means how to 3. That means if you decide to move through 3 steps, how many different ways you can go? Okay. So that's the idea of so-called Kazu index. Kazu index is to compute 1 step. This is this is adjacent matrix. That's what the network description. So that's 2 steps. That's 3 steps. You can have more. Then you put them together. This, add this, all together. This, add this. That's what you described. So if you add all this together, to the infinity, okay? So if you put them all together, that's so-called Kazu index, so in the network centrality measurement. So the summation of this, the whole thing, it's a global communication potential or power from one node to another. You can move directly, you can move through multiple steps. Okay, so this describes the overall connection between nodes. Yeah, so this is an example that you want to do detailed analysis of your network, and so to show the importance of nodes, you may have to use multiple different centrality measurements. In this example, if you only rely on degree, you will see, okay, looks like this gene are most important, those nodes are important. But if you decide to use closeness centrality and you get most important gene are here, and if you use harmonic centrality here, if you use Kazu index, the focus should be on here. So each gene or each node, you can use multiple, you can use 4, or you can select more different to describe this relatively important compared to others. And every let's see, month or every years, there are many new index will be proposed. If you look at the journals, for instance, I just randomly pick one as an example. It's called leverage centrality, compute this shared neighbors or whatever. Okay, so you can read. Authors just come up with ideas to get some unique features in terms of topology of the nodes to describe or amplify those relationship or computation. That use different ways to compute, and you can have a unique uniquely designed, such as leverage centrality. You can always come up with your own idea. Okay. Any questions before we move on? So, as I said, this is important because this concept will help you later. There's a lot of other things are related to this. So, let's describe the multiple steps, the connections. So we will quickly move to WGCNA. So this is a method called weighted gene co-expression network analysis, developed in UCLA. That's Professor Steve Harvus, very early 2008. Implemented in our package, as I said, has been widely used. There are multiple steps. So first, the input will be the gene expression profiles of multiple samples. At that time, there's no single cell data. It's just a table of the genes with different conditions or samples. So it's not the cells, they're samples. So samples could be from a different time measurement, could be from different conditions, experiment conditions. So that's input. X, okay, you have genes. Then conditions. Or samples. Okay. So to us, samples are cells. This will be hundreds, okay, thousands, easily get a simple experiment. But before single cell become available, you can hardly get many samples. Okay, you can consider I use mouse number 1, mouse 2, maybe different animals, collect samples from a different time. There's week 1, week 2. So that's one condition you can have. What else you can have? You can have, yeah, it's hard to see, okay? So you have those samples maybe from a wild type, and then those samples from a condition, knockout. So you have basically 2 categories. So I show you this is we never get enough samples before single cell become available. You can have 4 replicates here, 7 replicates here, whatever. The number is never enough. The most important is, in that case, you have only 2 conditions. You want to compare this condition against that. So in reality, you only have the shift of a condition. You only have 2. So you try to use this shift to construct a network from multiple genes. That's impossible. So I would see most of this co-expression analysis suffered from insufficient sample size. Okay, so if you have, see, I measured in 1,000 animals, mouse model, and their genetic variation in there. So there's a long table, have a mouse in different conditions, and then I can start to measure whether these 2 genes co-expressed across those genetic variations or experiment conditions, temperature, everything. See the variability is okay, but in most of the case, you have something like this, okay? So for the green genes, green group genes, you have high, low at the same time. But this basically just two conditions. So the link you can make, the correlation coefficient you make is underpowered, because by random you have a lot of those genes in this field condition will go up and down at the same time. So I would say never trust any results from a WGCNA, okay, unless you know it's have enough samples to measure the small variability in that. Okay, so if you look at this graph, you have 3 red genes, they all go up at the same time in the case control, and you put the link between them strongly, and then there you have 3 genes as a green group go down. Yeah.
When you say samples, do you mean number of cells?
I would see if in a single cell, yes, number of cells.
So the more cells, the more the
Yeah. But remember, this method has been used thousands of times before the single cell data become available. So I have no idea how people study only 2 conditions, see, control case, and try to infer a network between so many genes. So everything from this analysis is supposed to be underpowered, or there are many false positives. But anyway, people just use this to build co-expression network, okay? So once you have co-expression network, then you have a correlation. So correlation is between -1 to 1. So this is a range. So this method have to work on the non-negative values. So they did a transformation to take every absolute value. So don't consider even the negative. So everything is positive. Or you can do something like this: 0.5 plus 0.5 times correlation coefficient. So because this correlation coefficient is between -1 and 1, so once you have this transformation, and your range becomes from 0 to 2, okay? So this transformation allows you to make this whole co-expression network. Any negative values become something, and between the range between 0 and 2. So that's the deal. Then you have the fully connected network. Then you need to have a cutoff. See, anything greater than a value. So you need, for example, you have to anything greater than 0.7 will be considered. So R has to be greater than, so any this correlation greater than 0.7. So you select the cutoff, and you keep the link. Otherwise, you just remove them. You can also use some kind of this log. This transformation to take power of a value of beta. So some kind of soft threshold. Do a transformation first. So anyway, so you get a network by training this co-expression network. But my criticism for this is your co-expression network not accurate, not meaningful. So anything below doesn't make any sense, but people just keep using it. It's okay. So once you have this network, and you transform using this method into a non-negative weighted network, then you do this one more transformation. It's called topology overlap matrix transformation. Okay. So this method will have co-expression matrix. This become a network. Then you do one of the transformation and become a Tom. So transfer co-expression network. This is n by n. Okay, genes. You will do a transformation. This will be the same dimension, still n by n. So everything is the same size. So this is matrix, okay. Co-expression matrix, the transformation. And then the pipeline will do clustering, okay. Or use a simple tree-based clustering to group genes into clusters. Basically, that's all analysis is about. So now we are going to look at how this step has been proposed, what they are doing here. I would say this will be a kind of a smoothing, okay? Transformation to merge and smooth things. So this is called gene TOM0, okay? So you can see, okay, this is a before transformation. It's called TOM0 power. TOM0 is the co-expression matrix. So these are the transformed correlation coefficient. TOM1 will be this topology level of smoothing by consider the link between nodes, between genes, one step, okay? And then later there's extension which make this power 2, so which is considered 2 steps to merge. So this so-called topological overlap measure, TOM. Use this as approximation. So if you have 2 genes in the network, they have a shared topology. They overlap a lot. This will help them to merge. So that's what I said. This is kind of smooth things out. So smooth based on the topology position or similarity between genes in the network. Okay, their idea was from another paper. It's called Hierarchy, Organization of Modulity in the Metabolic Network. So this is from a Barbalassi group in the Spanish group. They published this paper in 2002, and to consider the so-called topological overlap. And Hoover has their group just borrow that. So this UCLA group just borrow that idea and apply to gene expression, co-expression analysis. So that's where the topology overlap concept come from. And this is how this topology measurement is computed. Let's consider, so you have I and J, okay, so put this position. There's 2 genes, and the correlation is there. So I and J. So now we compute topology similarity of I and J based on this network. And for I for itself, so this location will be 1. Okay, so that's where if I and J are the same gene to itself, you have topology level overlap 1. So in this position on 1. And then they consider the neighbors, okay, the number of neighbors of I. So if you have a neighbor, one-step neighbors from gene I, and the number of these neighbors, and you have a shared neighbor with the J gene, okay, also one-step neighbor, and you put them together and count how many shared neighbors, okay, and then plus this whether your I and J are linked, then you just add this value, this weight here. If not linked, you have a 0, right? Then you found which number of neighbors are the smaller. Okay, you have a neighbors for I and J, and you found the minimum, and then do that. So that's the way you compute the value in this Tom matrix. So basically, this number tells you how their network neighborhood of itself, nearest neighbors, overlap to each other. Okay. So these two may not be directly connected, but if you shared a lot of neighborhood, then you have a strong link between them. So that's what I mean, smooth things away. So there's some examples. I don't think we have time to go through. I can quickly show you this, for example. So let's see how this is computed. So topological values in the network A. So this example in A, and the link between node 1 and 2. Okay, these two. So they have shared neighbors. Two shared neighbors. These are the two shared neighbors. So that will be a 2. And they don't have a link between them, so this will be 0. Okay? And then for each of them, there are 4 neighbors. For 1, you have 4 neighbors. For 2, you have 1, 2, 3, 4 neighbors, right? So the minimum for this is 4. 4 plus 1 minus 0. So that's a denominator, and on the top you have 2. So 2 over 5 is what I compute this. Is it correct? 0.4, right? So that's how you compute each pair of this so-called topological overlap value of measurements. Okay, as I said, this Tom 0 is unchanged. So this is a given co-expression matrix. And then you can do one-step smooth and compute topological overlap measurements by step 1. So this gave you a matrix look like this. Look like this, okay? So if you allow 2 steps, so each node you consider 2-step neighborhood, and you get a smoother smooth matrix, okay? Looks like this. And then you can do higher power and topological measurement 3, and that's what you get. Okay, so after you finish this transformation, then you use clustering algorithm. So this tree-based, you can apply to the standard. Standard means TOM1, okay? Then they propose to use TOM2, okay, to do the clustering and get a better topology similar modules for the genes. So you can do the topology, you can do the clustering directly on this before transformation, but the results are not good. This after smoothing and it become better, and this year okay, you have a power 2 smoothing, you get an even better one. The larger modules can be detected. So that's what they're doing, the module detection through this clustering algorithm. Okay. Then they have given each cluster or module a color name, so different name. And then for those genes, you can compute some summary statistics, and if you have samples information as well. So that's WGCNA is doing. Okay, I quickly mentioned modularity detection before our break. So that's for a large network, you can consider their sub-modules, okay, or sub-network. So within the sub-network, nodes are densely connected to each other. Those network may be, modules may be linked by those hubs, okay, intermodular hubs. And then you have within the hub modules, you have central nodes. And that's what a lot of this biological network looks like this, okay? So it's interesting if you have algorithm can detect which nodes cluster together form a sub-module, and how can you separate them clearly. And so those are the algorithms people developed for community detection, okay? So just like in the social network, you want to find cluster of the peoples. Here, this just use these same algorithms to apply to gene regulatory or co-expression networks to find communities of genes. And then those community may contain the genes share the same function, or they play the similar roles maybe at network levels, okay? There are many different ways to do this community detection algorithm. And this, I will come back to explain this Lovin modulity, okay? Yeah, take a break, come back in 5 minutes. Okay, so this modularity computation, the key is in this so-called delta function here, okay? So this function indicate is like indicator. If you have I and J, the two nodes belong to the same submodules, okay, because we are detecting modules. Then you have a value, say 1. If C and J are not in the same module, have not been assigned to the same module, then this data function can have a different values, could be 0, okay? So there's a high contrast between whether two nodes belong to the same module or not. So this is a data function will be here, and then you compute these values, okay? This weight, the linker, and then so put everything together. So not only compute between I and J, just two nodes, but overall every pairs of the computation. Then your assignment of the community, this will be really matters, and then you try to maximize the Q value at the end. So that's your solution. So you will see this kind of equations in many of these modularity detection algorithms later. So use this technique, we can see, okay, we can design a test, not only differential expression, so something like this, but also differential modularity change. So in one condition, you have very strong modules, but if you, under another condition, this module is disappeared or increase or decrease. So you can start to compute something like that. So this idea in this methods which try to detect the change of modularity level before and after treatment. Now the single cell comes in and we solve the problem of those, okay? So I'm really concerned of this. The more people use this method, I become more concerned because everything you spend time, energy, money, fundings in there, you got the wrong information. And most of it should be wrong information. So now we have using single cell data to provide enough samples, okay? Because each individual cell is just some kind of sample, and each sample differ from each other a little bit by different reasons, okay? Could be the development stage, could be the random noise conditions and the technique, but you create some variability among them. If you have two genes always vary at the same time across all thousands of cells, this gives you strong indication they are functional linked. So I would trust the network inferred from single cell data more. As we said, we have 10x genomics data like this, and if you have a correlation compute across individual cells, cell pairs, gene pairs, you can do something like this correlation or regression. Have you seen this before? Yeah? So which lecture? I showed this before in one of these lecture before. So each dot here is a cell, and these two genes look like positively correlated. Real one increase expression in those cells, then AICDA have a high expression. So normally you don't see this beautiful regression relationship because the sparsity, as I said, they all zero, a lot of zeros. So all the zeros will be collapsed into here or here. So this data has been imputed using magic, okay, in order to show the relationship between correlation. If you look at these two genes, there's some nonlinear relationship between there, okay? So single cell data allow you to delineate this nonlinear relationship. For example, if you look at the third gene, PRDM1, so this color indicate the yellower and the higher expression of this gene. So those cells have a relatively high expression level of PRDM1, so this group. So if you only look at those DRPM1 highly expressed cells, then these two genes are positively correlated. Well, in the cells that this PRDM1 expression level is low, so those blue cells, and then these two genes are negatively correlated. So if you consider 3 genes, you start to use single cell data to construct this conditional correlation, right? Conditional. So these two genes correlation is conditional on whether PRDM1 is highly expressed or lowly expressed among the cells. So if you consider those relationships, and you can almost reconstruct the whole regulatory network. So this example is from NF-kappa B regulatory network in the B cell. Okay, so for the antibody secreting B cells in their differentiation regulations. So those are the how many genes? So this is 1, 2, 3, 8 genes in this network. So this 8 genes by 8 genes correlation matrix. So what we see before was this AICDA versus RIF4. So you see this nonlinear relationship that's between these two genes, and you can see the pairwise scatter plot for all the pairs genes. So from there, and you can infer positive and negative correlation, and then reconstruct this small network. And all these solid lines are the network which we can infer directly from the correlation coefficient. If it's positive, then you put this as a positive. If negative, then you have a negative inhibition. And most of these are correctly inferred. Only this and that made it wrong. So this is a PRDM1 to RILA. So PRDM1 to RILA, which is supposed to be, yeah, so it's here, okay. So this one is missing. Yeah, so the dash curve are the one which missed in the traditional previous study. So anyway, so this idea is to show you that we can use single cell data to reconstruct the network, okay? Because it provide a lot of these samples, so samples become enough. The example I showed you here is using a homogeneous population of cells. This is a B cell derived cell line. So there's no structure, no cell type. But in reality, you can build cell type specific networks. Okay, so some of this linker is only for green cells, and some only for the brown cells and the purple cells, this blue cells. So everything you can do can be cell type specific. So this is another layer of the advantage of the single cell data applications in the gene regulatory network construction. So you can do cell type specific analysis. And you don't expect the network is the same from one cell type to another, right? So you need to have that. So advantages, you have thousands of cells, sample size is large enough. And so within the cell populations, there naturally occurred variability. You have to have variability in order to do the correlation between genes. If there's nothing changed, you don't have any power to infer anything. But the single cell data provide this natural variability. So you can do everything cell type specific, of course, and you can have pseudo time or real time theory. So you get the population of cells and also cross time. So you do have a very strong power to detect two genes or gene pairs co-evolve or co-expression at a different condition, different time among the population of cells. So that's an advantage. So this is another review paper, and so you can have dynamic network inference because you have time theory, you have a pseudo time, you have trajectories, you have clusters, different cell populations, different cell type or cell in a different state. So you have those compared to just one static network. So single cell really provide a lot of advantages. So this is the idea to have your pseudo time trajectory built and along for this temporal information to add into the network inference. So you can see, this happened in the blue stage of cell and the yellow in the middle, and then the topology are changed. So I will go through several software tools, methods which have been developed for single cell gene regulatory network inference. So so those tools have been benchmarked and in this study. They try to use a standardized framework or the standardized input data. They set up this called a D-line evaluation framework. And all these methods either in R, Python, or Julia. So they have to set up a framework which can incorporate all these different methods into the same, under the same umbrella of this, to run this, to test on the standard gene set. So this is the method they used, and they are accuracy measurements, and all this, and whether it's computationally efficient, whether you need to have pseudo-time information, for example, SC code, and need a pseudo-time, so that's a tick here. And gene 3 does not require time series data, so it's cross. So this is a review paper. They test the accuracy of inference using 4 different networks. The problem with this test is all very small network. For example, the first one is only have 5 genes. The largest one is about 20 genes. Okay. So when we talk about inference of gene regulatory network, we talk about thousands of genes, say 2,000, 5,000. There's no ground truth, and really difficult for any evaluation method to say, okay, my method is better than yours, because in biology you can hardly get a ground truth. But here the authors just use this 4 model networks, and they say, okay, these are the ground truths. Okay, So your method need to infer network as close as is ground truth, as a golden standard to evaluate your accuracy. So that's accuracy, and you can get from a different method. So as I said, this is a small network, and this is the largest. GSD have about 20 genes. And they all 50% or slightly high in one of these methods. Now let's talk about the Genie 3. So this is a method proposed in 2009 for so-called decision tree-based method. So at that time, people probably set up those kind of test set, and they have a competition called Dream 4, Dream 5 to let people to submit their method and see which one, using this blended test, which one can get the most accurate inference of network. So Genie 3 is one of the top algorithms has been developed so far, and the winner of this competition. It's a tree-based, based on the decision tree. So we will quickly introduce the decision tree, how the tree can be decision tree like a binary trees, right? How binary trees can be used in this case to infer the relationship between genes. So any of this you need to consider whether these two genes are linked or these two genes are not linked. So they use decision tree-based method to have this so-called learning set to do things, okay? So let's jump to the decision tree. So decision tree algorithm is called a classification or regression tree or CART. So the CART algorithm is the foundation for important about this decision tree development method and the random forest. So use a lot in the classification field in the machine learning, right? So you can use CART method to do classification as well as regression. So this is an example for the binary tree model. So to do this classification. So yes, no, yes, no. So you have a lot of this yes, no binary decision boundary, and to classify your samples and into, in this case, male and female. So you can visualize this by some kind of this decision tree, what we should call this, partitioning map. Or then you have x, the x1, x2, and then you have cutoff, and put x1, put the samples into different bins. Then x2, you can do the same thing. And then gradually, you have a different level of the classification for the y. So this is the visualization of the decision tree. So you have training data to set up this boundary to cut. Then this landscape basically fixed by your training using the training data. So this is an example. This to do the regression, because if you look at this, the car powerhouse and their wheelbase, they are correlated. So you can separate those decision boundary around those training data and set up these cutoffs, and then you can do the regression for any given powerhouse. You can produce the values for this based on which bin you jumped into. So as long as you have enough the boundary to separate the points, so you change the classification algorithm into a regression because you have enough classes, and then where the Y is located in which class become a value, because you have more classes, become a regression. So the Gene 3 is to using a random forest method. So to deal with this gene expression matrix, okay? So you have a genes, and then you have an expression in different samples. So this is experiments. So this work, how it work? So you that's your input. Then you isolate one of the gene out, and then use all the rest of genes to make a tree. So I will isolate this gene first out, and then use all the rest of G1 as a matrix to predict gene 1 expression. Okay? So just leave this one out as a target variable you want to predict, then build a decision tree regression to use all the rest of genes expression to predict the first gene's expression. And then you move to the second gene. So this, each of these is called a learning set, and then you build a decision tree or regression trees based on the regression method. Then you put all this together and to construct the ranking, and then to do the prediction, to set up as a network. I don't know exactly how this ranking step is doing, but that's the idea. So it's using this tree-based, random forest tree-based method to do this prediction, then rank this gene, then make a network. So because this method is so accurate, okay, in the before the single cell egg. So now people also use this to single cell data as well, okay. So it seems it's really robust enough to deal with this sparsity in the single cell data. You have a lot of zeros, right? So far we believe this method still works very well in the single cell world. The only problem is too slow. It's because you have to repeat many this decision tree random forest for each gene, and then they have a sample. So people develop this parallel computing method to make this faster, but still take much longer to compute compared to co-expression matrix. This is very quick, right? Then there are other methods like a so-called scenic, and it's using Gene 3 as its core to build a network. Then add another steps. For example, after you calculate the gene regulatory network, then they start to search. Okay, search transcription factor and the target database, and keep only known relationship between transcription factor and their target. Because you create a network by algorithm, and some of those are not a real relationship. It could be co-expression because the two targets under the same regulation, they look like linked, but there shouldn't be a direct relationship between regulation between two genes. So scenic just do one more step, or cover up or a filter to remove those unknown relationship between transcription factor and target. Only keep this relationship already registered in the database. The database they use is a cis target and iregulon. So those are the experiment validated transcription factor and the target relationship. and they use the gene 3 create this O2O network, then they start to cut. If there's no known relationship between this regulation, I will remove you. I only keep the last most important ones. So that's the step. So they use gene 3 to build network, or use the boost method, which make the gene 3 faster. Then they keep only regulons, which known relationship between TF and the targets, and they remove those non-registered regulation relationship. And then once you have this, they only consider those target genes and the relationship they can compute. So within the cell population, under this particular relationship, so those target genes, which cell have a high expression of those target genes, so means those cells under the regulation of these transcription factors relatively strong compared to another. So you can use this relationship to classify the cells, and some cells have this gene regulation strongly imposed on this. Some cells are not. So there's just one more step to use the cell score to compute that. So the cynic is just a workflow and nothing new. It's just a borrow different idea put together, and has been widely used since then. And people use this to develop downstream annotation, and also found the binding sites, because all these genes, gene 1, 2, 3, 4, under regulation of the TF1, in front of those genes, they may share the motif, and you always see these signatures shared among those. So you have a very strong indication that they're directly binding from this transcription factor, because you know the binding site is the same of this consensus sequence you found in the target. So you can make very beautiful stories biologically. Okay? There something else, and people use information theory, so to do this so-called partial information decomposition. They borrow this mutual information computation and then put them together, similar to the partial correlation. And then there are other methods to use differential equations to utilize this time series data. Okay, so you need to have a pseudo time assigned to the individual cells to compute a network. People also start to consider to have so-called causal. Okay, so if you believe this network next step, so time theory, so next time the expression or relationship rely on the previous time. So if you have a network from time 0 and then for time 1, so this relationship, if you can find this regulation cross time, then you can infer the causality. Okay, so that's a really powerful approach to see this potential regulation and push the network move. And so people start to think about the multi-layer network method. You can do modularity detection at a different time than to find the change of modulity. Okay, it was here but not there, and reappeared in the time 3. So those methods use the Louvain, for example, the algorithm I told you, including this time theory information, and make it really complicated. So that try to find the network switching, okay, multi-layer network switching. So people use time varying graph. So the graph theory method we can borrow So some of this method may not developed for single cell data, but if you know those methods, you probably can borrow them and directly apply to single cell data, because we can easily construct single cell data time series network, something like this, which normally require a lot of efforts to collect those data, right? Their method based on the non-negative factorization. So this is have a time series network, try to find the modules, which features, which shared across time. Okay, so you need that essential module across time all the time. So this method can detect those consensus modules, basically. Okay, very quickly introduce what we have done. So we try to construct a gene regulatory network and also compare. Okay. Have I mentioned this before? No, not yet. Okay, so this is what we try to build: two gene regulatory network from two population of cells. It could be condition 1 against the 2. We try to find the genes. Looks like a gene 2. Okay, gene 2 strongly correlated with 6, 5, 4 in this condition, but it changed. It become weaker in condition 2 or this 2, and make a stronger link with gene 1. So we believe if we can compare these two networks and find those genes, this change its regulation. This is called a differentially regulated genes. Okay, remember we have a differential expression, so differential variability. Now we can use this method to detect differentially regulated genes, networkally regulated genes. So this is a method called single cell 10-foot net for constructing and comparing single cell gene regulatory network. So we will have two samples, one expression matrix, another. You can do some subsampling to create some subsamples in order to control some of these outlier cells, okay? Because if you subsampled and average them, then you remove those outliers. So here we use a regression method called principal component regression to construct the network. The idea is you found one gene, found a pair of genes. Okay, you compute the regression. Regression means gene 1, gene 2. Okay, you want to build a regression curve, and to get the slope, right? Y. T. So you want to get the slope. Slope is your beta is in your correlation coefficient. Similar as a cointegration network. So, but the PC regression is not only consider the correlation between G1 and G2, condition on all the rest of genes. So all the genes without the G1, without the G2. So this is a kind of partial correlation between these two genes, and they remove the effects of all the others. So all the others will be using a PC, okay? All the others will be use the PCA basically to get the top PCs and put in there as a covalence, as a control variables, and then you compute the correlation between G and G1. So we found this method is fast enough and also accurate enough in the GSD test, okay, in the B-line test. So you can construct networks, and then because it's a multi-dimensional, become a tensor, so you can use tensor decomposition and keep top components and reconstruct, make a denoised version of this, and then we can align them, consider them as a manifold, and you do this manifold alignment. Then look at the same gene because they share the same gene. You have a black version and the red version of the same genes. So, but after after aligned into the same manifold, then you can compare the same version, same gene A in the black version and red version, whether they are well aligned. If they're well aligned, they will be close to each other. The distance between black and red version will be small. If you have a gene which the distance is large, that means you cannot align them very well in terms of network alignment. Then those genes will be picked out, so they are not alignable because the two networks change their topology. These genes cannot be aligned very well. They are differentially regulated in their respective blue and black and red network. So those genes will be picked out and the differentially regulated genes, and it go enrichment analysis. So we will play this in computer session to see, okay, if you have a sample, something like this. If you do DE, then those genes, there's no difference. There are multiple animals, 4 in the red group, blue in the 4 in the treatment group. So those are the data from the morphine injection in the mouse neurons and the neurons expression. There's no difference at all. So differential, this analysis is really good if you have two conditions. Differential expression cannot find anything significant because the cell population before and after treatment are so similar to each other. The only change is very subtle dysregulation, just like this. All the genes are at the same level, no expression change, no significant average expression difference between two conditions. If you have that experiment data, then apply this. What you will find all those genes which change their regulation power or relationship with other genes in the network. So those genes will be fished out using the method I showed you. And those top genes, if you look at their function, in this particular example, they are morphine addiction, opium signaling, G protein signaling pathway, the gamma AB receptor activity. So they all related to the challenge in this mouse experiment. So this is a very powerful approach, okay, which cannot be done because the challenge is short term. The gene expression has not been changed or modulated substantially, so you don't see the expression level difference among the important genes. So this is a so-called pseudo deletion, so it's called a virtual knockout. So same pipeline, but we just not using the so in this case we will be compare wild type against knockout. So that's what experiment design, mostly knockout experiment will be doing. But our virtual knockout is you don't need knockout samples. You just have a wider type expression. You can download a lot of wider type expression matrix, and you make a copy. So you copy yourself. So if you have two identical matrices, you can still run this. You will generate some random genes because there's no difference. The whole pipeline will generate noise. You still get the results. But what we will do is to set one of the gene into expression level across all the cell becomes 0. So this simulate a knockout effects. So after you manually remove this gene, you can run this pipeline again. You will get a list of genes. So those lists of genes, the gene you set into the knockout will be always on the top, the most different ones. But the second, third, will be interesting genes. Those genes are if you knock out this gene, this particular one, those genes will be impact regulatorily. Okay, so this is an example. We knock out TRIM2 genes from the mouse neuron, and we can predict after using this, using wider type information for the microglia, and then you knock out the TRIM2, and you will get the genes which in this function groups, Alzheimer's disease, lots of them, all this will be significantly impacted. So those are the results. And we compare this with the real knockout animal data. So that's a paper the original author published. They also get those pathways, function group. If you compare this with a virtual knockout, they're almost the same. So that means you don't need to do this animal knockout experiment. You can just virtually simulate the knockout of this microglia, and you get exactly the same biological significant pathway or function about this gene. Okay, TRIM2 genes, if you stop its function in the animal microglia, you will get response of those genes, and those genes are responsible for the Alzheimer's disease or those lots of them. If you do the animal experiment, you get the same results. So you don't have to do it. Okay, basically that's the idea. Yeah, to replace animal study. Yeah. So we will play with this Thursday and see how you can set up to make predictions. So the idea is you have a good network built from the single cell data. It's accurate enough that any perturbation on the network, so any place you will be able to locate and what the impact, where the impact is. So I already mentioned this XCT, so this also gene regulatory network based. So their network here. Okay, so I think that's all for this week's lecture, and see you Thursday, and hopefully you get some good idea of network analysis later. Okay. Let me know if you have any questions. I will send out the presentation list later this week. So 9 of you. So I believe we need next Tuesday and Thursday 2 days to finish the presentation platform. Okay. So we'll keep you posted and give you a list, and we'll let you know who's going to give a talk next Tuesday, who's going to give a talk next Thursday. Okay? We do. I think Tuesday's talk will be here, and the Thursday will be there. We still keep doing this. Yeah.
Then what about the
week follows, we can we haven't decided yet. You can have 1 week break and prepare your test, or we can have an immediate test. So it depends on you. Depends on which you want to finish quick or right after week 14. So next week will be week 14 for the presentation and the discussion. Then if you want to have a test week 15, we can come back to this. Or you want to do week 16 and we still come back to this room. So we can decide. Okay, I can want to have 1 more week to study. Okay.
Thank you.